Okay. So as an introduction, we are going to look at the cell, cell communication and the pathways. And then in the evening, we are going to look at the receptors and the pharmacology now, the different receptors, and how we'll revise the pathways because we know the drugs act through the receptors. Am I breaking? I'm being told my internet connection is unstable. So, I'm breaking. Let me restart my Wi-Fi.
Yeah, you're breaking.
So the first part, I'm sure you're familiar with this because Seraphine did this when we were looking at introduction to biochemistry. That is the cell. So the cell, who can remember the cell organelles and what their functions are? Because once you mention those, we'll be done with this first part, the cell. You can just mention any cell organelle and what function it does from this diagram. Or rather the major parts, the plasma membrane. We know plasma membrane surrounds the cell, and it's the medium through which any material desiring to enter or leave the cell has to pass through, so that it can either enter or leave the cell. And we know the plasma membrane here is made up of the lipid bilayer. Lipid bilayer, let's see here. Okay, lipid bilayer is nini. We know how it looks like. Let me draw. There's this, the hydrophobic head, and then the lipophilic tail. Then here, like this, yeah. That's why we call it the lipid bilayer, because it's made up of two layers. This part is hydrophilic. Sindio? The head is hydrophilic, and then the tails are hydrophobic. And you can see the tails make up the bulk of the lipid bilayer. So that's why we say something has to be lipid soluble in order to diffuse through the lipid bilayer, or to diffuse through the plasma membrane. And on this point, all cells, now we are talking about humans. We're talking about humans. All cells have that plasma membrane. They have that lipid bilayer. And then the other thing is that specifically in humans or in eukaryotes, even the other organelles are also surrounded by the membrane. Now, in that case, it won't be called the plasma membrane. It will be called, maybe like the mitochondria, will call it the mitochondrial membrane, but it's still made up of the same lipid bilayer or phospholipid bilayer, because it's made up of those phospholipids. Yeah. Then for the other organelles, we have the ribosomes. Ribosomes, they are responsible for protein synthesis. We have mitochondria. Mitochondria here is energy production. We have the endoplasmic reticulum, that is the smooth and the rough endoplasmic reticulum. The difference is that in the rough endoplasmic reticulum, we have ribosomes attached to it. So the rough endoplasmic reticulum will also be responsible for protein synthesis, same as the ribosome. The smooth endoplasmic reticulum, on the other hand, the reason it's called smooth is because it doesn't have these ribosomes attached to it. So that's why it's called smooth endoplasmic reticulum. And for it now, it functions for the packaging of the proteins. And then its function is closely related to the one for the Golgi apparatus or Golgi complex, which will also function to not only package, but also modify the proteins that are made from the ribosomes. And then the lysosome, you know, for destruction, we have the lytic enzymes there, destruction of any harmful materials that are inside the cell. The nucleus is the home of DNA. There's DNA synthesis, RNA synthesis will occur in the nucleus. And surrounding the nucleus, again, we have the nuclear membrane. So it's what we said. In eukaryotes, we have even the cell organelles being surrounded by a membrane, being enclosed in a membrane. Unlike what we saw in the prokaryotes, the bacteria, which there is no cell membrane surrounding the organelles. Okay. So we've talked about this rough synthesizing of the proteins, smooth packaging. Yeah, packages new protein in the vesicles, and the vesicles will transport them to the appropriate place. We've talked about the lysosome. Though all of this is a revision. Okay, the one thing we didn't mention in biochemistry was about the proteasome. And yeah, the proteasome is where misfolded proteins are broken down. They are broken down to amino acids. The amino acids, you know, will just be recycled by the body. And so that a protein can be broken down inside the cell tags it with this molecule known as the ubiquitin, which is a small protein. Just ubiquitin, once anything, if it's an organelle, when a protein is tagged with ubiquitin, or when a protein is attached to ubiquitin, that is a death sentence. It is destined for destruction. And where is it destroyed? In the proteasome. I think that's the one place we didn't mention in biochemistry. This, you attach a protein to ubiquitin, and it will now be destined for destruction. Yeah, in the proteasome, not in the lysosome, in the proteasome in this case. Yeah. So for the Golgi apparatus, again, just modifying of the proteins, and also now it will sort and segregate according to their function. So this is now the packaging of proteins and secreting them to the right place. Packaging, secret, yeah. This. Yeah. I'm moving fast in this place because we've done this. Lysosomes, we know for lysosomes is about destruction. Lysosome means just to break down or to kill. Then let's look at how stuff from outside the cell is internalized into the cell. We know this process is called endocytosis. Endocytosis, endo means inside. Cytosis is kind of movement towards. So endo into the cell. Exocytosis is movement outside the cell. So for endocytosis, we have three major ways through which endocytosis occurs. So endocytosis is a broad term that describes how stuff is moved from inside the cell, from outside the cell into the cell. So endocytosis is a broad term. So for endocytosis, there are three ways through which it can occur. That is pinocytosis, whereby in pinocytosis, we'll see majorly it's fluid related, but we'll see that receptor mediated endocytosis. Now, from the word receptor mediated, it means that first there is binding of the substance to a receptor, after which it is somehow it's enclosed into a cell and finally it enters phagocytosis. For phagocytosis, we saw it when we were talking about immunology. So to summarize this, we'll just look at the diagram so that it's easier. Yeah. So let's start with pinocytosis. In pinocytosis, you can see, first of all, this is the cell. Now we are seeing water molecules and solute molecules are outside the cell. So simply in pinocytosis, what happens is that the membrane pockets will just pull inwards, enclosing this stuff. And now after being enclosed, there's that endocytic vesicle that has been formed, and that is it. You've enclosed your material into your cell. That is pinocytosis. It's that simple. There's your water molecule and solute molecules there outside your cell. The cell membrane pinches off that stuff, and then it is internalized inside the cell. So pinocytosis is just that straightforward. And we'll say it's kind of related to phagocytosis, kind of. Yeah. Now let's see receptor-mediated endocytosis. For receptor-mediated endocytosis, when we see the word receptor, we know that there has to be something binding to a receptor. There has to be some kind of specificity to it, because we know receptors are specific. So we see the target molecule here, and these are the receptors, the stuff in blue. Yeah, these are the receptors. Now the target molecule will bind to the receptor, and then once it binds to the receptor, we'll see the membrane pocket will pull again. The process is the same. It's just the kind of the first step, which is different from pinocytosis. It will pull inwards, and then we'll have the endocytic vesicle with the target molecule inside, which has now been internalized. But with this, we'll see this clathrin molecule, which coats this vesicle that has been formed. We'll see the details of exactly how these things take place just in a minute. I just want you to see the diagrams first. Yeah. Now, finally, for phagocytosis. Phagocytosis, you can see the prey. Imagine this is your bacteria here. And now this is your macrophage or your neutrophil, the phagocytes, right? So the phagocyte will form pseudopods. The pseudopods, they will extend covering this area of the prey. Then once they extend, once they've extended and they've covered the whole prey, they will attach to each other now, which will ensure that this E. coli, this bacteria, has now been internalized. And then something unique to phagocytosis is after it's been internalized, this stuff is called the phagosome. It's internalized inside the phagosome. Then the phagosome will now fuse with the lysosome to form the phagolysosomes. And we know in lysosome, we've said it has lytic enzymes or enzymes that kill. So once it's internalized inside the lysosome, or rather in the phagolysosome, the bacteria or the toxin, whatever it is, it will be broken down. Yeah, it will be broken down to so that it's no longer harmful to you. So let's go to the details slightly. So pinocytosis, we've seen it's as simple as what? This. Just a reminder, this. Cell, water, the membrane pinches off, and then you have your stuff. It's literally this, okay, cell drinking. where a droplet of the extracellular fluid is taken up non-selectively. Remember, in receptor-mediated endocytosis, for that one is selective. The target molecule has to bind to a specific receptor so that we are able to internalize the specific target molecule. But now for pinocytosis, it's just non-selectively, the cell will be taking a droplet of the ECF. Okay, so in detail, the plasma membrane will dip inwards CF. Then at the end of the day, we'll have it in an endocytic. Am I breaking? I'm being told again, it's unstable.
No.
Okay. So yeah, it will be enclosed inside that endocytic vesicle or endosome. Then what in receptor-mediated endocytosis, we saw that there is one molecule that stands out, and that is CLA3. Now, in pinocytosis, the one molecule that stands out is dynamine. This protein or this molecule is the one that will be responsible for the pinching of that endocytic vesicle. Okay, so in pinocytosis, it's what we've described here. It's just what is here, the cell pinching off a drop of ECF and enclosing it or internalizing it inside the cell. So the only other information that we are adding here is what protein mediates this? It's dynamine. It's the protein that mediates pinocytosis. So pinocytosis is mediated by that dynamine protein or dynamine molecule. Then for the second one, we have receptor-mediated endocytosis. In this place, we've seen for wherever there is this word receptor, because we'll be talking about receptor a lot now that we've come to physiology, pharmacology, we'll be talking about receptors a lot. Wherever we are seeing this word receptor, we know that it is very specific. Unlike pinocytosis, whereby it was just taking a drop of ECF, it can be anything outside the cell. But for receptor-mediated, it's targeting something very specific, something that can bind to the specific receptor and now be internalized. So hence, it's a highly selective process that imports specific. Again, the key word here is just specific, that it needs from the environment. So the key word here is just that word specific, because it is receptor mediated. Okay, so it will be triggered when that target molecule, such as the protein, binds to that specific receptor. Once now that binding has occurred, the stuff will be internalized, or it will be brought inside the cell. And that internalization of the specific protein, the specific target, is formed, is mediated by this CLA3 molecule, which lead, or rather, which mediate formation of that pouch, that vesicle. You can call it that vesicle. Yeah, it's mediated by CLA3. Yeah, which these are membrane deforming cord proteins on the inner surface of the plasma membrane that will bow inward and form to the outward curving cord that form buds. The resulting pouch is known as coated pit because it is coated with CLA3. So let's go back just to the diagram once again. You're seeing CLA3, here it is. Here is the plasma membrane, here is CLA3. The receptor, the molecule has been bound to the receptor now. Once it has been bound, now we have activated the receptor-mediated endocytosis, and we see now CLA3 will now form the outer covering. Okay, it will now form the outer covering of the endocytic vesicle in this case. Yeah. Or coated bead, clathrin coat can also be called the clathrin coat. Yeah. We can see what can be absorbed through this cholesterol complex, vitamin B12, hormone insulin are selectively taken into cell by receptor mediated endocytosis. Remember, for vitamin B12, it has to be absorbed, meaning the intrinsic factor has to be present so that vitamin B12 can be absorbed by a cell from the GIT. So in this case, what is recognized by the receptor is that intrinsic factor. That intrinsic factor is what is essentially, it's what leads to that absorption. So if you ask, well, how is vitamin B12 absorbed, it's through this receptor-mediated endocytosis. And what is the target molecule? It's intrinsic factor. That's why if you don't have intrinsic factor, you can't absorb vitamin B12. Yeah. So a clinical note is this: even viruses that attack us, they use the same mechanism. We know HIV, right? HIV, we know it has the receptors. We talked about the CCR5, CXCR4, the CD4. It utilizes the same mechanism that we utilize to gain access into the cell. Yeah. Finally, phagocytosis. Here, simply, it's about the white blood cell will, these pseudopods, they just extend. It's like the cell membrane extending to hug the bacteria, just here. For phagocytosis, is just the cell in this case is the one making the effort to hug the bacteria. So this is your macrophage, this is your neutrophil. It sees this E. coli, it sees this bacteria molecule, whatever it is, toxin, it extends. Like, imagine the pseudopods as hands extending to hug the prey. Once it hugs the prey, it encloses it into something called a phagosome. The phagosome, the phagosome will fuse with the lysosome to form the phagolysosome, where the bacteria will be broken down, where it will be killed. So in this case, for phagocytosis, at the end of the day, all of this in pinocytosis, phagocytosis, receptor-mediated endocytosis, the stuff will first be enclosed in an endocytic vesicle, right? It will be enclosed in this endocytic vesicle. Even this one is just an endocytic vesicle, and that is a coated one. But now in phagocytosis, it's still called an endocytic vesicle, but the other name for it is now a phagosome. Yeah, because it's phagocytosis, it's got this phagosome. Phagosome fuses with lysosome, and now we have phago. And now we'll have phagocytosis taking place. Now we will kill the bacteria. For phagocytosis, we know majorly, or not even majorly, principally is just for for the immune cells. Yeah, it's principally for the immune cells. So we see here as a note that most body cells will perform pinocytosis. Also, many will carry out the receptor-mediated endocytosis, but only a few are capable of phagocytosis. And we know the most important of these are the white blood cells or the immune cells, the likes of macrophage, neutrophils, yeah, the B cells. Then now we proceed to, regarding this, the function. I think we've all seen these things, unless you see something that you want us to cover specifically, but we've talked about these cell organelles, mitochondria, glycolysis, TCA, Nini, the electron transport chain. We talked about this in detail when we were looking at Nini, biochemistry. The TCA cycle, I hope you remember. The mnemonic, let me see if I can. The mnemonic was just for the TCA cycle was citrate is Krebs starting substrate for making oxaloacetate. Citrate is Krebs starting substrate for making oxaloacetate. So that's how the molecules of the TCA cycle follow each other. And Nini, we know the first step in TCA is acetyl-CoA will combine with oxaloacetate and will form citrate. Then that's why we say citrate is Krebs starting substrate for making oxaloacetate. Yeah, I hope you remember that mnemonic. And the acetyl-CoA is formed from pyruvate. Where does the pyruvate come from? It comes from glycolysis. Glycolysis, where does glycolysis come from? From glucose. Glucose, where does glucose come from? From carbohydrates, breakdown of carbohydrates. So when you eat that ugali, when you eat that rice, broken down to glucose, glucose comes to glycolysis. From glycolysis, we obtain pyruvate. Pyruvate is transformed to acetyl-CoA through the pyruvate dehydrogenase complex. Acetyl-CoA can now enter the TCA cycle to produce more ATPs. Yeah, to produce more ATPs. Electron transport chain we covered, so yeah, these details, unless someone wants us to just go through it again, I'll just be skipping through it. Yeah. So this is a summary of all the cell organelles with their function. Okay. Yeah. And we've talked about, yeah, all of this. So next we are going to go into signalings. We'll take 1042, I think, not 1042, 1044. Yeah, 1050, we can go through cell-to-cell signaling. We can take a break if someone has something to add, something to, or rather a question, Nini. Yeah, you can address it now. At 1050, we'll be going to cell-to-cell communication. I'm going to write the TCM mnemonic on the group chat. Yeah.
Guys, is this textbook on the drive?
Honestly, I'm not sure. Let me,
Can you just send it on the group,
yeah,
please?
let me send it on the group.
Thank you.
I will. I hope the break was refreshing. Now we're looking at cell-to-cell communication. How do cells communicate with other cells? Okay. And again, I'll just work with a diagram first so as to explain the simpler stuff, and then we can go to the technical details. Okay. Now, the cell-to-cell communication, it can either be direct or it can be indirect. It can either be direct or indirect. Direct is the least common, or rather indirect is the most common form of communication between cells. Indirect is the most common, not the direct. Direct, not so common. It happens, but it's not so common. Indirect, most common type of communication between the cells. Okay. So for direct communication, as the name suggests, it means that there is no medium between the two, that these two cells are actually communicating with each other. And it happens through two main methods. Number one is the gap junction. Now, for the gap junction, is simply that these are two adjacent cells. Let me draw another here so that it's also yeah, these are two adjacent cells that have literally a gap junction between them, so they are able to communicate with each other. There's a gap junction between them, so the cells, they are adjacent to each other, but they have a junction connecting the two. It's like you have your own home and your neighbor also has their home, but then between your fences you put like a small gate that you can use to be communicating between the two of you instead of you having to go out, nini, and all that. In short, it's like a small gate between two neighbors, right? You have your compound, I have my compound, we have this fence, but in the middle of the fence, there's a gate that we can use to communicate with each other. Okay. And how will cells communicate? The communication is not verbal, it's more through transfer of various chemicals. It's just through the transfer of chemicals. Then the other method or the other mode of direct communication is this transient link up of cell surface markers. Transient link up of cell surface marker. Transient means just short-lived, that through some cell surface markers, two cells are able to communicate, and then the link up is just short-lived, and then the cells go their own separate ways. And in transient link up here, majorly we are talking about the major histocompatibility complex, whereby, or okay, just anyone who can say anything about MHC, major histocompatibility complex. I don't know if Stanley is here because it's majorly an immunology thing. Anyone on anything on the major histocompatibility complex? How many are they? How do they work? You don't have to be really, you don't have to mention all those technical terms. Okay. Tough crowd. Tough.
Gaia.
Divya.
Okay. According to Google, I've just Googled it. It says the function of MHC molecules is to bind peptide fragments derived from pathogens and display them on the cell surface for recognition by the appropriate T cells. Yeah, that's all. And then yeah, so there are two types. There's MHC1 and 2. They play a pivotal role in the adaptive branch of the immune system. Yeah, that's all.
That's all. Yeah, essentially that is it. The major histocompatibility complex is just a presenter. It presents the molecules that are inside our cells to the external environment. It presents them to the, and now in the external environment, in this case, we are actually meaning to the T cells. That's what the major histocompatibility complex does. It presents our internal, or rather our cellular antigens to the T cells. And as Divya has just said, there are two types. There's the MHC class 1 and MHC type 2 or class 2. Okay. Now, in this case, to better understand, or rather, before again moving to the complex explanation of what the major histocompatibility complex is, let's start with the basics. My cell has been infected with a virus. How will my immune cells work to make sure to identify that this is an infected cell and this one is a normal cell? We know, remember what we said about viruses. Viruses, they have to enter inside our cells and then they make their own proteins, right? These proteins, they are not, they don't look identical, or they are not similar to my proteins, okay? And that will, and the major histocompatibility complex, of course, through it functions using the Golgi apparatus. We'll see how it functions later, but essentially it will be able to display that there is a foreign protein inside this particular cell. And then the T cell will come and be like, hey, this one is displaying a foreign protein. And what the T cell will now do, it will kill off that specific cell, okay? So simply the MHC presents what is inside the cell to the T cells. The MHC presents what is inside the cell to the T cells, both the CD8 and the CD4, okay? And so we've covered an example of virus. A virus-infected cell, inside the cell, it will have viral proteins. MHC will display those viral proteins. And then once it displays them, the T cell will come off and kill that cell. Then for another example is cancer. In the case of cancer, in cancer, we know simply put, cancer is a mutation, in that there's been a mutation, or it's simply a mutated cell, or rather the DNA has been mutated, leading to production of proteins that are also mutated. So you have your normal proteins, and then you have this mutated protein. So the mutated protein may, in the case of cancer, let me say the word may, it may not look similar to your normal protein. Then the MHC will pick that up, present it to the T cells, and the T cells will be able to recognize this is a cancerous cell or this is a mutated cell. And the T cell will be able to kill off that particular cancerous cell. And now in the development of this immunomodulating anti-cancer agents, that is essentially what they are targeting, such that your immune cell is able to mount an effective immune response against the cancerous cell. Yeah, but now for cancer, we're not talking about cancer, but for cancer specifically, it has a lot of tactics that are able to evade the immune system. One of them being, it's just able to mimic your own proteins. It's able to mimic our own proteins. That is one of them. And the other thing, it's also able to turn down the immunity. So this whole process may end up even not occurring. Yeah. So to nini to cover the MHC because you all refuse to unmute now. I have now to cover the MHC, but I'll summarize. The MHC, there's class 1 and class 2. There's class 1 and class 2. For class 1 is found in all cells. Okay? Class 1 MHC molecules, it's found in all cells. Class 2 MHC molecules is found specifically in immune cells that, or rather, not even in just immune cells, in antigen presenting cells. Class 1 MHC molecules is found in all cells. It's found in your liver, it's found in your lungs, it's found everywhere. Every single cell in your body has class 1 MHC cell. And then class 2 MHC cells, it's only found in the, or rather on the antigen presenting cells, because the MHC is found on the cell membrane. So I'll say it's found on the antigen presenting cells. Which are these antigen presenting cells? We cover them: the dendritic cells, the macrophages, and the B cells. Those are the antigen presenting cells. And among them, who can remind us, among the three, dendritic, macrophage, and B cells, which one did we refer to them as professional? You know, among the three, which one do we say it does the best job in antigen presenting? Anyone? Okay,
Yes,
for this
I
one,
don't know, but is it
uh-huh.
dendritic? Is it dendritic?
Yeah, dendritic. Yeah. Nice one, Divya. Dendritic. The dendritic cells, we call them, these are the professional antigen presenting cells. Professional meaning they do an exceptional job as compared to the macrophages and B cells. Okay. Yeah. So class 1, we've said they are found in all cells. Class 2 in the antigen presenting cell. And then the other key difference, I'm not going to look at all the differences, I'm just going to look at these two key differences. The other key difference is that in class 1, the mediator or the inspector, the mediator, and finally the executioner, should that cell need executioner, it will be the CD8 cells or the cytotoxic T cells. But for class 2, the mediator and the inspector is the CD4 cells. It's just the CD4 cells, the T helper cells. Okay. So class 1, we said they are found in all cells, and the mediator is a cytotoxic T cell. That means it's as simple as that. My cell is infected with a virus, cytotoxic T cell inspects the MHC, it finds this foreign antigen, and it injects the mediators to kill this cell, right? The granzyme, the perforin. We mentioned all of that in immunology. Okay. But for class 2, we've said it's found in antigen presenting cells. And the mediator in this case is the CD4. Remember even what the CD4 is called, the T helper cells. The T helper cells, what does it do? It will ensure, it will produce the cytokines to mount an immune response, or to help, okay, to help in now further action of this antigen. Okay. Yeah. So that one is just because we had this, that is what now essentially, I know I've taken you all the way and then now I'm back here. So that is what this other type of direct cell communication is about, in that there is a short link up of cell surface markers. In this case, we said is what it is, the MHC presenting what is inside the cell to the T cell. The T cell inspects it. If it's fine, the T cell moves on. That's why it is short-lived. It's transient. If it's fine, the T cell will move on. If it's not fine, it's a viral infected cell, it's a cancerous cell, the T cell will kill it off. Okay. Yeah. Then now we go to the indirect methods of cell-to-cell communication. For the indirect methods of cell-to-cell communication, number one, what we've said about this is that this is the most common way by which cells communicate. Okay, this is the most common way by which cells communicate. Direct, nah, not so much. Indirect, this is where the magic happens. And in indirect, now we'll see there is this paracrine secretion, neurotransmitter secretion, hormonal secretion, and neurohormone secretion. Paracrine here, we are going to tie it also with something called autocrine. Okay. Now let's start with paracrine though, which is the one here. Paracrine, it just acts locally. Paracrine, you just okay, a good example, let's say paracrine is what happens when let's say people are engaged in cheating of exams. Okay, it's for example, there's this cell here that will say that the answer to number 1 is B, and that answer will just go around the people seated around this cell. So that is paracrine. That's why you hear, you guys don't cheat exams. That's why you hear when people are cheating exams, you hear this section chose number 1 is choice B, the other section choice C, the other section choice D, because the communication is local. It's just to the surrounding environment, not just to the neighbors, but to the surrounding environment. It's like in paracrine, think of it as more of this cell, and then there's a radius through which it will communicate to this other cell here, other cell here, other cell here. Okay, so paracrine is just to the neighboring environment. That communication it will give off, it will just go out to the neighboring environment. Okay, then on this, let's also mention autocrine. Auto, the word auto we've seen over and over again. Auto means self. Autoimmune disease. It means the immunity is attacking itself, right? Autonomic nervous system. It means it drives itself. It doesn't need your intervention. Automatic vehicle, it drives itself, Yeah. So autocrine is whereby the cell communicates to itself. Remember, in cell-to-cell communication, at the end of the day, what is happening is that there are mediators. There is chemicals that you can see, these red chemicals. They will move from A to B. That cell, we say they don't communicate through words. They will communicate through molecules, through chemicals. So in paracrine, you've seen this chemical from the secreting cell will go out to the neighboring cell. Okay. Then in autocrine, the secreting cell will release this, let's call it this green stuff. It will release it, and then that green stuff will just act on it. Okay, it won't act on any other cell. That is what autocrine is, that the cell will release this green stuff, and then that green stuff just acts on it. Okay, yeah. So that is it about paracrine or autocrine. Okay, paracrine to the neighboring environment, communicates to the neighbors, to the neighboring environment. Autocrine to self. The cell is essentially just signaling itself. Okay. Then we go to neurotransmitter. For neurotransmitter, there's nothing much to be said there. There's a neuron, it will secrete a neurotransmitter that will act on the cell. The target may be a muscle, it may be a gland, right? It may be anything. So for neurotransmitter, that one is that simple. No, nothing much to be said there. Okay. It's just for example, now for neurotransmitters, just acetylcholine binding to those muscarinic receptors, nicotinic receptors, adrenaline being released, or noradrenaline and binds to the adrenergic receptors to the other side. Yeah, that is just it. Neurotransmitter is simple. I'm sure that one doesn't need a lot of stuff. Hormonal, also the same thing. Hormonal is now what is in hormonal, will have the secreting cell will release the hormone into the blood, and the hormone can go from the brain all the way to wherever it's going, right? It can go very far. We know for paracrine secretion, because we said it's just to the neighbors, it's very local. Okay. An example of paracrine secretion or paracrine communication is when there is inflammation and there is a release of histamine. We only see dilation of the nearby blood vessels. There's only that dilation there, but the vessels that are not in the injured side, they do not dilate. Your aorta, your what, it won't dilate. It's just, for example, if you've hurt your finger, it's only the vessels around that specific finger. Even in the other finger, those vessels will not dilate. That's a good example of paracrine. It's just neighboring, just short. For neurotransmitter, we know neurotransmitter that the signal can come from your brain, but go all the way to your legs. Okay, so it can be long, but the mediator at the end of the day acts locally. That's the message can come from far, but the mediator will act locally. We know the acetylcholine will just act at that particular synapse. Okay, if that synapse is at the leg, it will just act there. It won't. So if I want to contract my hands, the signal will come all the way from my brain, yes, that I want to contract my hand, but the acetylcholine will be released between my nerve at the hand and my muscle at the hand. That is where the acetylcholine will be released, and that is where it will act, or rather, that is where we will see an effect. So it can, so for the neurotransmitter secretion, the message can come from as far as possible from the brain all the way to the legs, but the chemical mediator that will ensure or that will bring out an effect will just be local. That contraction will just occur in my hand, despite the message of coming all the way from the brain. For hormonal now is very it's a what? This one can travel the hormone where the hormone is released and where the hormone will be acting. It can be 1,000,000 miles away. And a good example of this is something like insulin. Insulin is released in the pancreas, but insulin can work all the way to your legs, right? To the muscles of your legs. Another example is the hormones of the pituitary gland. For example, the FSH. FSH will be released in your pituitary gland. That is essentially in your brain. But it will enter your bloodstream, go all the way to your ovaries, to your testes to work there. So in hormones, what we are seeing is that the mediator is released into the blood, and then it will act on the relevant site. How does it know this is the relevant site? The relevant site will have receptors for it. Insulin will have the insulin receptors for it. Okay. If it's FSH, the same. FSH receptors will be there. If it's the TSH, the same. Okay. That's hormonal. Yeah. Then finally, we have the neurohormone secretion. The neurohormone secretion. Now, just as the word suggests, it's that there's a neuron component, or there's a there's a nervous component and there's a endocrine component. So neurohormone secretion. What is unique is that the mediator in this case is released from a neuron, hence the word neuro. But the mediator goes to the blood, okay? And it can act at a distant site, hence hormone. So why neuro? Because the mediator is released from the neurons, okay? But it travels through the blood to a distant site to act, okay? That's why we say it's a neurohormone, okay? The mediator is released from a neuron, enters the blood, and acts on a distant site. A good example of a neurohormone here, it's simply, again, well, let me not say again, in this case is the posterior pituitary glands. Remember, anterior pituitary glands, we call them the adenohypophyses, meaning it's a glandular cell. But for the posterior pituitary gland, the other name for it was a neurohypophysis, meaning in posterior pituitary gland, for example, ADH, vasopressin is released from the posterior pituitary gland. And the posterior pituitary gland is made up of neurons. So the posterior pituitary gland neuron will release the ADH into the blood, and then the ADH will go all the way to your kidneys so that it can act. Now that is an example of a neurohormone secretion, that the mediator or the chemical is released from a neuron, but it enters the blood so that it can reach its target site. That is a neurohormone, okay? So the difference between hormone and a neurohormone is that for a hormone, the mediator is released from the glands. We know glands are the stuff that produce these hormones. So for hormonal secretion, the mediator is released from the glands. But for neurohormone, the mediator is released from the neurons. But at the end of the day, mediator travels in the blood to the distant target site. Okay. Yeah. And I believe that's it. That is it for cell-to-cell communication. Unless someone has a question or something to add on this, we're not going to go through the statement, or rather this part that has been written here, because I think I've explained it in the diagram has covered what we need to do. So anyone has a question or an addition, please can feel free to step in.
Guys, so there's not much difference between neurotransmitter and neurohormone, apart from the hormone getting transported to the blood, and for neurotransmitter to be targeting the local target directly, right?
Yeah.
Okay.
Yeah. No more questions or additions. So just to summarize, okay, there's even no need to summarize, but because the diagram has summarized. The most common way by which cells will communicate is this indirect communication. Okay, this indirect communication. Direct, not so common. Okay, but for direct, the two types, gap junction, and then we've seen that MHC link. Indirect, paracrine, neurotransmitter, neurohormonal, and hormonal.
I have a question. I don't know if it's a question or a contribution. You talked about major histocompatibility complexes, and you talked of class 1 and class 2.
Yeah.
And we saw the CD8 working on class 1 and the CD4 on class 2. But remember the CD4 as T helper 1 and T helper 2, TH1 and TH2. And I can't remember, but there's either TH1 responsible for infections, but TH2 responsible for antibody production. I'm not so sure, but there's a slight difference on how, yes, they do by class 2, but there's a TH1 responsible for certain for certain conditions and TH2. Please, if we can check on that, but I'm not sure. I can't remember.
Yeah, you're right. Yeah, TH1 majorly is the infection, both virus and bacterial. Then TH2 is production of the antibodies. And that's why we see TH2 even functioning with what's these things called? Allergic reactions, because IgEs mediate allergic reactions. That's why TH2s are for antibody production. Yeah, you're right.
Thank you so much.
If there is no more addition at 11:30, we are going to look at the pathways. Now, we've mentioned the cell has secreted its stuff, the chemical. In this case, we call this the green stuff. The green stuff has gone, it has binded to our receptor. What happens then, right? Even the hormone has gone, it has binded to our receptor. What happens then? Yeah, so we are going to look at the different pathways through which now that communication will be transducted or will be followed up. Yeah, so we can take the 6-minute break. 11:30, we start on the pathways. So one last subtopic, and then that will be it. So as a revision, we said that cells communicate with each other. We say that communication can either be through direct methods or direct communication, or indirect. For direct communication, we've seen the major type. There is a gap junction. That's the major type. And then for indirect, you've seen there are these 4 types. Paracrine communication, whereby we are talking about neighboring cells. Okay. Neurotransmitter, don't have anything much to say there. We know about neurotransmitter, and we're going to look at this when we're talking about CNS physiology and pharmacology. Hormonal secretion, we talk about insulin, estrogen, wherever hormone, any hormone you can think of falls under hormonal secretion. And then for the neurohormone secretion, here we're talking about a chemical mediator that will be released by a neuron, but will enter into the bloodstream and work at that distant site. How is neurotransmitter secretion different from neurohormone secretion? Neurotransmitter, we have the chemical mediator being released into the synaptic cleft, where it will act directly on the target organ, right? Neurotransmitter will be released into the synaptic cleft, where it will act on the target organ or on the target tissue, wherever it is. But for neurohormone, the let me not call it a neurotransmitter yet. The chemical mediator will be released into the blood and act at a distant site. That is how neurotransmitter is different from neurohormone. Neurotransmitter, synaptic cleft, neurohormone into the blood, different distant site. Then how is hormonal secretion different from neurohormonal secretion? For hormonal secretion, we have the chemical mediator is being released by a gland or an endocrine cell, right? It's being released by a gland or an endocrine cell. So in this case, we have a simple example of insulin. Insulin, that is hormonal secretion. We have a gland releasing it, that is it's the pancreas, or if you want to get, we need the beta cells, right? Which count as a gland. The endocrine portion of the pancreas releases insulin and acts at a distant site, okay? But for neurohormone, we have the mediator is being released by a neuron, but enters into the blood and acts at a different site. The mediator is being released by a neuron, but enters into the blood and acts at that distant site. Example of neurohormone, we've seen the ADH. ADH is released by a neuron in the posterior pituitary gland, but it enters the blood and acts on the kidney to mediate water reabsorption, okay? So that's the difference between neurotransmitter, hormonal, and neurohormone, because this neurohormone, it combines the two of them, okay? Now, we're looking at the pathway. How we've released this insulin, we've released this ADH, we've released estrogen, testosterone, whatever mediator, we've released it. How does it work? Okay, how does it mediate its function? And again, for this place, for signal transduction, or now it's called signal transduction. For this signal transduction pathways, or the pathways which the cell will use, the mediator will use, kindly, let's pay attention. Let's pay attention, because when we are going to talk about the drugs, we won't be repeating the same things. We'll just be saying it acts through the G stimulatory pathway. Because now today we are going to look at these pathways in detail, so that when we are saying something like salbutamol acts on the GS receptor, or rather beta 2, sorry, not beta 1. Beta 2 is a G stimulatory receptor. That's all we'll be mentioning. We won't go again and look at how the G stimulatory receptor works. So please pay attention on signal transduction. So signal transduction, see, just from the name, it means that there's an incoming signal, right? In this case, we know it's a chemical mediator, right? That's the incoming signal. It will come to my cell, and then transduction. Transduction means I will kind of not transmit, but I will kind of interpret, right? And lead to another thing. It means the signal will come, for example, let's say insulin. Insulin will come and say, the blood sugar is high, we need to absorb more sugar into our cells, right? That's the message insulin brings, that the blood sugar is high, we need to absorb, or we need to mediate the entry of glucose into our cells. That is it. That's the work of insulin. That work is done. But now in signal transduction, now we will see how insulin has come and told us we need to take up sugar from the blood. Now in signal transduction is whereby we can see how will we take up sugar from the blood. Okay, so that's what we are looking at here, and we are seeing this is very close to how drug works, how drugs work. Yeah, the same thing. Drug will act through these pathways. Okay. So before talking about how a signal will eventually bring about a desired response, we'll look at the chemical messengers or the chemical mediators. The chemical mediators, they are divided into two types, right? And that is just based on their solubility. There's a lipid-soluble ones and there's a water-soluble mediators, okay? Just simply based on solubility, lipid-soluble and water-soluble. And when we were starting off, when we were talking about the cell, we said the cell is surrounded by a lipid bilayer, right? A phospholipid bilayer, a lipid bilayer, a plasma membrane. And we said this plasma membrane is lipophilic or hydrophobic, lipophilic or hydrophobic, right? So if something is lipophilic and the plasma membrane is also lipophilic, that will mean this stuff doesn't need a lot to enter that cell. Bringing you back to chemistry, like dissolves like. Or rather, in our case, for pharmacology, for physiology, we know if a drug is highly lipid soluble, it easily diffuses from the blood into the cells because it's highly lipid soluble, easily diffuses. Okay. Now that's the same mechanism, or rather that's the same method that Nini will use, the lipid-soluble chemical mediators. If a chemical mediator is lipid-soluble, it will just, for example, a very good example of the lipid-soluble is the reproductive hormones, the likes of estrogen, testosterone. Those are very good examples of lipid-soluble chemical messengers, right? Estrogen will be released by your ovaries and it will just enter your bloodstream. And once it enters your bloodstream, it just diffuses into the cells, okay? We've never talked about there being an estrogen channel, okay? Or an estrogen, I don't know, pump that pumps estrogen from outside into the cell, right? That's because estrogen is highly lipid soluble. So you know estrogen has that cholesterol, what is it called, nucleus, okay? That means it's highly lipid soluble. So it simply diffuses from your blood into your cell. And now for the lipid soluble messengers, the receptors are found inside the cell. Right? The receptors will fall inside the cell because this stuff easily diffuses from your blood or from your ECF inside your cell. But for the water-soluble messengers, for the water-soluble messengers here, because for this water-soluble messenger to enter into my cell is very difficult, we see that the receptors will be found on the surface of the plasma membrane. That is where the receptors will be found. So that this water-soluble just will act on that receptor and then the receptor will know what to do. Okay? Yeah, because for this mediator to enter inside my cell, it's really difficult. It's really difficult for it to diffuse through my lipid bilayer. So it just acts on the receptor on the plasma membrane, and then it leaves the job to the receptor to make sure that the desired outcome, the desired response is achieved. So for signal transduction, or rather for chemical messengers, we have seen two types. The lipid soluble, which will easily diffuse through your lipid bilayer and then bind to the receptor inside the cell. And then for the water soluble one, it won't diffuse across your lipid bilayer, so it will act, its receptors will be found on the surface and it will act on the surface. It will act, or rather it will bind to the membrane receptors, and the receptors will be left with the heavy responsibility to achieve the desired response. Yeah. So now we can go to the pathways. We've already talked about the types of messengers. Now we can go to the pathways. Now for the pathways, here we have pathways now for the lipid soluble and pathways for the water soluble. We've already said lipid soluble, we have the receptors are inside the cell or they are intracellular. But for water soluble, the receptors are on the surface membrane receptors. Okay. So for lipid soluble, we have intracellular receptors. For water soluble, the receptors are on the surface. And for lipid soluble, we said the steroid hormones, those who each have that cholesterol structures, are the ones that are majorly function through this mechanism: estrogen, testosterone, progesterone, even cortisol. You know, cortisol is still a steroid hormone, right? They function through that mechanism. And what's common about testosterone, estrogen, progesterone, once they bind to their receptor, that we call it that hormone receptor complex, or rather hormone receptor it's called element. Whatever it is, we'll see that later. That moves to the nucleus and alters gene expression, right? Alters gene expression, bringing about now the changes will now be brought about when the gene expression has been altered. You've promoted synthesis of some proteins, you inhibited synthesis of other proteins, hence, and that will bring about those changes. That's why for even something, a guy to change to a chick, it takes time. You know, you can't just wake up and take your estrogen and you become a chick. No, because estrogen has to go to the cell, it has to go to the gene level, alter transcription of your genes, make sure that you are not synthesizing genes that are making testosterone, or rather you're not synthesizing proteins that promote the male gender or a characteristic of the male gender, right? So your larynx will no longer be thick. You'll start developing breasts, all those things. Yeah. So in short, these steroid hormones, you'll see the actions take time. But with recent research, just to mention on this, with recent research, they've been found there are also surface membrane receptors for these steroid hormones. Okay, just note that there are also surface membrane receptors for steroid hormones. Don't assume that all receptors are found in the intracellular environment. There are also surface membrane receptors for these hormones. So there's some aspect of them having receptors on the surface membrane. Remember, for something like Addisonian crisis, right? For Addisonian crisis, to save that dude, you have to inject hydrocortisone. And if we are saying hydrocortisone has to go diffuse, act on the gene so that the gene expresses some protein, that takes time. That person will have passed on, okay? So that's why you're saying there's also some surface membrane receptors that mediate some very few specific functions. But the major functions that the steroid hormones mediate, they function at the cellular level, at the gene level. Okay. Yeah. Now the pathways for the water soluble ones, we already say these are the surface membrane receptors. We can have these chemically gated receptor channels, or we can have these channels that bind to and activate receptor enzyme complexes, And finally, and the most important that we're going to focus on is the G protein coupled receptors. We know the G protein coupled receptors, this is the heart of pharmacology. So this is where we're going to put our efforts on. We'll just mention the others, but majorly we're going to take our time with the G protein coupled receptors. Yeah. So far, if there's any questions, you can ask before we go to the now any type of receptors. I know the discussions will end at 12, but kindly just give me until maybe let's say 12:15, because the G protein we just have to take our time there so that in the evening we can do pharmacodynamics. Okay. So we won't mention the intracellular receptors, or rather the lipid soluble messengers, estrogen, testosterone. We'll see them when we're talking about reproductive physiology and pharmacology. Yeah. So now we'll look at the water soluble messengers. Okay, and we've seen they can act by 3 major mechanisms, or rather, the first one is the messenger can bind to a chemically gated receptor channel. Okay, a chemically gated receptor channel. Once it binds to such a channel, there'll be ion movement leading to a cell response. So based on this explanation, who can tell me an example of an example of a messenger or a chemical mediator that acts through this mechanism, whereby the messenger binds and leads to opening or closing of a channel, resulting in ion movement? Anyone? Can be a drug, can be anything. Can be anything. In your 5 years of studying pharmacy, something that acts through this way. It binds it, it leads to either the opening or the closing of a channel.
Maybe I can think of drugs like benzodiazepines, which usually potentiate the activity of GABA by the chlorine channels.
Yeah. Nice, yeah. They potentiate the action of GABA. You know, when GABA binds, we open the chloride channels. Chloride enter the cell becomes hyperpolarized, so the cell is not easily stimulated. Hence, if you're treating epilepsy, you've essentially treated the cause of epilepsy. Yeah. Nice. Anyone else? Can be a drug. Okay, so someone give us a drug, but we've mentioned a physiological molecule. Now we want a pharmacological molecule. A drug that binds and either leads to opening or closing of a channel. Anyone? No one? For chat box, chat. An example is something like what digoxin. And yeah, we've seen digoxin when we were talking about pharmacognosy. Digoxins, it binds to that sodium potassium ATPase and closes that channel, right? Yeah, so that's another example, meaning a drug that can bind and it can either lead to opening or closing of a channel. Hence, we call it that chemically gated receptor channel. Chemically gated receptor channel. Okay. Chemically, while a specific chemical binds, for example, now let's say work with GABA. GABA binding, or rather GABA binding to that receptor either will lead to either the opening or closing of a channel. So GABA binding to the GABA receptors opens the chloride channels, and there's more chloride movement into the cell. Okay. Yeah. Then the second one here, we have the receptor enzyme complex, right? That's the other way, whereby now the messenger binds, the chemical mediator binds to a receptor enzyme complex. Why is it being called a receptor enzyme complex? Because this one functions as one, I wouldn't say as one at a unit or one molecule. It's a complex whereby we both have the receptor and the enzyme in that one complex. So the enzyme, the messenger binds on the receptor side. That binding leads to activation of the enzyme on the other side. And a very, the most common molecule that acts through this is insulin. Insulin is through this tyrosine kinase pathway, which is a good example of this receptor enzyme complex. Yeah. Yeah, it acts through this tyrosine kinase. Tyrosine kinase is that receptor enzyme. It means that protein, let me call it that protein that is on the cell surface acts as both a receptor and an enzyme. A receptor in terms of it will receive the mediator, which is insulin in this case, and an enzyme meaning it will start the process of ensuring the desired response. That's the tyrosine kinase pathway. And for the tyrosine kinase pathway, I know we've even seen the tyrosine kinase inhibitors. They, like, even just from the name, you can say they inhibit this. They inhibit this enzyme complex. Anyone who can give now an example of a tyrosine kinase inhibitor? I'm not asking for explanation. Example. Anyone. TKIs.
Erlotinib.
Erlotinib. Yeah, erlotinib. They have that suffix. Tinib, right? Imatinib, erlotinib. And majorly we saw them being used in which disease? There's one specific disease we saw tyrosine kinase inhibitors being the major treatments.
CML.
Yeah, nice. CML. The CML, chronic myeloid leukemia. Yeah. And why, or rather, okay. Wacha tusiende huko. But CML, simply put, in CML, remember we are having that is it what is it called? That thing? The translocation between chromosome 9 and chromosome 22, and that was leading to an upregulation of this, or rather that was forming this Philadelphia chromosome, essentially leading to an upregulation of this tyrosine kinase receptor enzyme complex. That's why we are giving the tyrosine kinase inhibitors. Okay. So yeah, And then the G protein receptor, which activates a second messenger pathway that will carry out the cell response. Okay, so we've seen three types. The first one is the chemically gated receptor channel. We've seen the likes of GABA. GABA is the chemical. Once it binds to its receptor, it opens the gate, or rather it opens the channel, chloride flows in. Okay, receptor enzyme complex, we've seen the receptor also functions as an enzyme. The receptor also functions as an enzyme. And finally, the G protein coupled receptor pathway, or GPCR pathway. Here, your messenger will bind to the G protein, and that G protein will lead to, or rather the receptor will activate other stuff that now will act as a second messenger to carry out the response. We call it a second messenger because the first messenger, for example, something like, let me say salbutamol. Let me take you back to salbutamol. Salbutamol activates the G2 receptors, or adrenaline. It activates the, not B2, yeah, the beta 2 receptors, right? And the beta 2 receptors, they're G stimulatory pathway. Or rather, that's their pathway. So adrenaline will bind to that beta 2. And then once adrenaline binds to the beta 2 receptor, they'll be, through a series of mechanisms, we'll have cyclic AMP increasing. And we say cyclic AMP is now the second messenger, because the first messenger was adrenaline or salbutamol. That was the first messenger. Now the second messenger is the one that will carry out the cell's response. In this case, it will be your cyclic AMP. Hence, we call it the second messenger. First messenger is your adrenaline. It's the drug, the hormone that started the whole process. Okay. Yeah. So for the chemically gated receptor channels, we are going to look at them more closely when we're talking about, again, the nervous system, because principally that is where they act. Okay. Now let's go to the tyrosine kinase pathway, and then we'll look at the G protein coupled pathway. Here's the tyrosine kinase. In the tyrosine kinase pathway, we said one thing. The receptor is also the enzyme, judge, jury, and execution. The receptor is also the enzyme. Okay. So you have here, and we said, what's an example of chemical mediator that acts through this pathway? It's insulin. Insulin acts through the tyrosine kinase pathway. Well, the receptor for insulin is a tyrosine kinase receptor enzyme. Okay. So insulin, it will come and bind to this site, the receptor site of the receptor enzyme complex. Okay. And there has to be two molecules so as to activate it. So one molecule binds to this side, the other molecule binds to this side. The binding of those two molecules, receptor enzymes protein kinase. Okay, it will activate the receptor enzymes protein kinase, and or rather, yeah, into brackets here, the tyrosine kinase. And here they are. So your insulin has been bound here. Okay, and then now your tyrosine kinase has been activated in the now cytoplasmic portion, or the intracellular portion. And then the tyrosine kinase will self-phosphorylate itself. Like this stuff self-phosphorylates itself. All right. Once it self-phosphorylates itself, it's now let's say, let me now say it's now truly ready to start acting, or now this is the protein that will now lead to your response. Okay, so the tyrosine kinase phosphorylates itself. Then secondly, it phosphorylates the designated protein. Once the designated protein is phosphorylated, it's now active and it's able to now carry out the cellular response. Okay, so insulin binds, and on binding of insulin, it activates the tyrosine kinase, which first of all phosphorylates itself. After phosphorylating itself, it phosphorylates the designated protein, and now the designated protein will be able to carry out its function. Okay, hapo tuko pamoja? Let me look at it. Okay. I was looking for a diagram. I'll find a good diagram for this, for insulin specifically. Yeah. Okay. So that's the tyrosine kinase receptor enzyme pathway. There's something unique. The receptor is also the enzyme, but the receptor portion is in the ECF, in the extracellular environment of fluid, and the enzyme complex, of course, will be found in the intracellular compartment. Okay. Then next, and now the most important one, is the G protein pathway. Okay, and we'll see why is it the most important. We are seeing most of these messengers will act through the G protein coupled receptor. They will act through this G protein coupled receptor. And for the G protein coupled receptor, the mechanism is the extracellular messenger. In this case, we gave the example of adrenaline. It will bind. Then on binding, the G protein will, one of its components will activate another protein. That other protein is now what will lead to the production of a second messenger. The second messenger eventually will lead to a cellular response. So in short, the G protein, it's complex, but it's the most common way used. So we have to now look at it. This is where our efforts should be dedicated. Okay. So first of all, let's just look at here, the clinical notes. Half of the drugs on the market, they act through this G protein coupled receptor. That's why it's like we can't escape it. Half of the drugs, they act through this receptor, okay? Because this receptor will participate in some way in most body functions. Hence, they are important for drugs. They are important targets for drugs. Yeah. So let's look at the general pathway, and then we'll go and look at now the two, or rather three types of G protein coupled receptors. There's the GS, the GQ, and the GI. GS, G stimulatory, GQ, I don't know what the Q stands for, and then GI, G inhibitory. Okay. So this is a general way in which they work. We have the extracellular messenger. For example, in our case, we've given adrenaline as our extracellular messenger. Adrenaline will bind, okay? And you're seeing there is this alpha, beta, and gamma. Now, always it's the alpha subunit. Once the receptor binds, once the mediator or messenger binds to the receptor, the alpha subunit, okay, will dissociate, okay? The alpha subunit will dissociate and travel across the plasma membrane to an effector protein. To an effector protein. The effector protein can be, in this case, let's say adenylate cyclase. Okay. Now the adenylate cyclase, in this case, just an example, will do what? It will produce the second messenger. We know for adenylate cyclase, the second messenger, it will convert ATP to cyclic AMP, right? And now cyclic AMP will be our second messenger, okay? The second messenger will act to, will activate a protein kinase. Usually it can be protein kinase A or protein kinase C. So we'll activate a protein kinase. That protein kinase is what will now phosphorylate the designated proteins, and these designated proteins are what will bring about the cellular response, okay? So let's go through it again. The messenger will bind to the G protein receptor. Binding of the messenger activates the G protein. On activation of the G protein, the alpha subunit dissociates, travels to an effector protein. For example, if you use adenylate cyclase, the effector protein will now be activated and it will produce the second message. Adenylate cyclase will produce ATP, will produce cyclic AMP from ATP, which now the cyclic AMP will be our second messenger. The cyclic AMP or the second messenger activates a protein kinase. The protein kinase will now phosphorylate the designated proteins, which will bring about the desired response, okay? Bring about the desired response. So that is it about the G protein. That's the general pathway, okay? So I hope we've all memorized it or understood it. Because now we'll go to the types of the G protein coupled receptor. Okay, we'll go to the types. Here we are. Just okay. So G protein pathway. Okay. Let's go here. We'll start with this one. The This is now the G stimulatory pathway, the GS pathway. Okay, we'll start with this one. Remember from the earlier diagram, messenger binds, activates the G protein, alpha dissociates, travels to an effector protein. The effector protein is what will make the second messenger. The second messenger activates a protein kinase. The protein kinase will phosphorylate now the designated proteins. Once the designated proteins are phosphorylated, we'll have our response. Okay, that's the general mechanism. Now we are looking specifically at the G stimulatory pathway, the GS pathway. Now, for GS pathway, number one, a molecule that acts through the GS pathway is adrenaline. Okay, adrenaline. So in our case, think of adrenaline acting. Not a molecule that acts on the pathway, on the GS pathway, a receptor that is a G stimulatory is the beta-2 receptor. Sorry, that was an INI, because the other types like alpha-1, for alpha-1, we'll see alpha-1 is a GQ. Okay, so beta-2, let's work with beta-2, because beta-2 is a GS receptor. Okay, beta-2. What acts on the beta-2 receptor? Adrenaline. So adrenaline has come. It's binded here. Binding of adrenaline to the beta-2 receptor activates the protein. It represents the G protein coupled receptor. Once it's been activated, we say the alpha portion will dissociate from the beta and gamma and travel across the cytoplasmic membrane to now this adenylyl cyclase. And it will activate it. This alpha subunit will go and activate this adenylyl cyclase. Activation of this adenylyl cyclase does what? Now the enzyme will function to produce cyclic AMP from ATP. So, the cyclic AMP from ATP. The cyclic AMP is what will activate our protein kinase A. Our protein kinase A. What will protein kinase do? Protein kinase will now go and phosphorylate the designated proteins, bringing about the desired cellular response. In this case, it will be something like bronchodilation. Okay. So again, G stimulatory, GS, messenger will bind, alpha will always dissociate. So after alpha dissociating, it will activate the adenylyl cyclase, which will convert ATP to cyclic AMP. Cyclic AMP acts as a second messenger, activating the protein kinase A, pKa. pKa will now phosphorylate the proteins that will bring about the desired response. So that is the GS pathway, the G stimulatory pathway. Okay. So yeah, an example is the beta-2 receptor. Beta-2 receptor is a GS. Beta-2 receptor is a GS. Okay. Beta-2 receptor is a GS. So let's go to GQ. GQ is where now in GQ we'll see calcium being our second messenger. Okay. Let's go here to GQ. So GQ, remember the first like two to three nini are the same. We said alpha-1 is a adrenergic receptor and it's a GQ. Okay. So the messenger is still adrenaline, binds to the alpha-1 receptor, which is a GQ. Okay. Binding again, activates it, the alpha subunit dissociates, and now will activate what? phospholipase C. Phospholipase C. Remember in GS, we were activating adenylate cyclase. In here, we are activating phospholipase C or PLC, okay? Activation of phospholipase C. What does phospholipase C do? It breaks down the PIP2, or in other times you can just say the membrane phospholipids, okay? Yeah, but okay, try to remember this, okay? This one, because I've seen this question. PIP2, it breaks down this PIP2 to IP3 and DAG, okay? Inositol triphosphate and diacyl glycerol, okay? Inositol triphosphate and diacyl glycerol. So phospholipase 2 breaks down, phospholipase C breaks down PIP2 to IP3 and DAG. Now, DAG will go ahead and activate protein kinase C, okay? For GS, we had protein kinase A, but for this, we are seeing DAG activates the protein kinase C. Protein kinase C will now phosphorylate the designated proteins, and the designated proteins will bring about a cell response. But that's not just it. Remember, we formed DAG and IP3, okay? We formed DAG and IP3. So DAG has activated protein kinase C. Protein kinase C has done its job. It has phosphorylated the designated proteins, and we've had a response. But what has IP3 done? Okay. Now on IP3, what it does, it will mobilize, or it will lead to production or release. The right word is release. Release of calcium from the sarcoplasmic reticulum or the endoplasmic reticulum, where calcium is usually stored in the cell. It will lead to this release of calcium from the, from the endoplasmic reticulum. Calcium now is another second messenger. IP3 is a second messenger, DAG is a second messenger. Calcium here is also functioning as a second messenger, okay? Calcium will do what? It will activate calmodulin, okay? Calcium will go ahead and activate calmodulin, okay? Then this calcium calmodulin complex, it goes ahead and activates a third thing, okay? And now this is the calcium, or rather, let me call it, calcium calmodulin kinase. That doesn't have to be deep. Just calcium calmodulin kinase, or CAM kinase. That calcium calmodulin complex activates the calcium calmodulin kinase, okay? Now this calcium calmodulin kinase, once we find a kinase, we know our job is nearly complete. It activates the calcium calmodulin kinase, and the calcium calmodulin kinase is what will now activate or phosphorylate the designated protein, bringing about a desired response, okay? So for the GQ is a bit more complex, but it's still kind of related. Yeah. Phospholipase, it brings about IP3 and DAG. DAG, protein kinase C. Then IP3 calcium calmodulin. Then calcium calmodulin activates calcium calmodulin kinase. Now the kinase is what will phosphorylate our designated protein, bringing about a cell response. In this case, usually it will be vasoconstriction, or rather just even contraction, okay? Yeah. Then finally, this book doesn't have a diagram of it, so I'm going to send a picture on the group, or let me see if I can display a picture here for the G inhibitory pathway. For the G inhibitory pathway, it's a very simple pathway. It's just not even the opposite of what happens with the G stimulatory. What the G inhibitory pathway just does, it inhibits the adenylyl cyclase. And we know once the adenylyl cyclase is inhibited, that is it. You've killed that pathway. Yeah. Hope there's a simple diagram here. Yeah. Yeah, this is here it is. Yeah, and it even summarizes all of them. So in the G inhibitory pathway here, we just seen a good example is the alpha-2 adrenergic receptor. In the G inhibitory pathway, what we have is simply the receptor, the messenger, in this case, noradrenaline has binded to alpha-2. Alpha-2 activates this G inhibitory. The alpha subunit will dissociate, go to the adenylyl cyclase and inhibit it. Once it has inhibited it, that is it. You know, it inhibits adenylyl cyclase. It's that simple. So to understand the G inhibitory, you have to first of all know that in the G stimulatory, adenylyl cyclase is the main stuff. Adenylyl cyclase, we have cyclic MP. Cyclic MP, we go and activate pKa. pKa phosphorylates the designated proteins. Yeah. So yeah, that is it from me. This picture summarizes everything. The GS, GQ, phospholipase C, we've seen from phospholipase 3, we have IP3 and DAG. DAG activates protein kinase C, and IP3, calcium. Calcium calmodulin, the complex activates calmodulin kinase. Calmodulin kinase will mediate the desired cell responses. Yeah. So if there's any question, any addition on this, it's welcome. Otherwise, that's it from me. I don't have anything else to add on this. If no questions, but we'll stop. We'll stop there. Then,
Could you please send that image to the group kindly?
Oh,
It wasn't a question
okay.
though. Thank you.
Welcome.